All right, group, welcome to Unit 17, where we're going to talk about gastrointestinal tract infections and stool cultures. Primarily, we're going to discuss Salmonella, Shigella, and Campylobacter. These are all included in what's known as a routine culture. Gastrointestinal problems are a very common patient complaint. About 300 million episodes of diarrheal illness occur every year in the United States, and there's about 6,000 associated deaths every year in the United States from gastrointestinal illnesses. So let's get started. The flora of the gastrointestinal system is established very early in life, and it remains relatively stable for life unless it's wiped out by antibiotics or some sort of disease process. Most bacteria are in the colon, but there are some smaller amounts that are in the small intestine, but very few organisms can actually survive the stomach, and that's due to the stomach pH being very acidic at about 1. Flora of the gastrointestinal environment would be things like anaerobes, enterics, non-fermenters, staphylococcus, and streptococcus. You can see here that in the upper small intestine, this harbors the fewest amount of organisms because it's closest to the stomach. The upper small intestine harbors about 10 to about 1,000 organisms per mil. And those organisms are usually streptococcus, lactobacillus, yeast, diphtheroids, and enterococcus faecalis. In the lower small intestine, that's a little bit farther away from the stomach, so we actually have more organisms. We have about 1 million to 10 million organisms per mil, and that would usually consist of enterics and Bacteroides, which is an anaerobe. And then the large intestine harbors the most organisms at about 100 billion organisms per mil, and that would include organisms such as the anaerobes, enterics, staphylococcus, and pseudomonads. So you might be asking yourself, how does our patient avoid being colonized with enteric pathogens? Well, for one, we have the presence of normal flora, and normal flora actually competes with pathogens for nutrients. We have the acidity of the stomach. The pH of the stomach is too hostile for most organisms. Shigella, however, can survive the pH of the stomach. Then we have normal peristalsis, which is the constant motion of the small intestine. We have a mucus layer where organisms are trapped or passed in the feces. And then finally, we have secretory IgA and phagocytic cells. We have secretory IgA, which is an antibody that is in secretions, mucosal secretions of the GI tract, and also secretory IgA is in the saliva and tears. And like I said, an infection of the gastrointestinal tract is a pretty common disorder, and there are a number of signs and symptoms that go along with gastrointestinal infections. Probably the most common presenting symptom is diarrhea, but that can also lead to dysentery, which is also severe diarrhea associated with abdominal cramping and blood cells in the stool. We also have fever that may or may not be present, dehydration because of the lack of fluids, and then also what's not on this slide are other symptoms such as nausea and vomiting. As you can see on this slide, there are a number of organisms that can cause gastroenteritis. Some bacterial organisms would be things like Shigella and Salmonella, E. coli, especially O157H7, Campylobacter, Vibrio cholerae and Vibrio parahemolyticus, Clostridioides difficile, Clostridium perfringens, Bacillus cereus, and Staphylococcus aureus. Also, what's not on this list would be things like Yersinia and Aeromonas. We also have viral causes of gastroenteritis, including norovirus, which is the winter vomiting disease, rotavirus, I had that, that's not fun, and astrovirus. And then we have parasites that can cause gastroenteritis as well, such as Giardia lamblia, Cryptosporidium and Isospora, and Entamoeba histolytica. The most common type of material collected for gastrointestinal samples would be fecal material or stool samples, and we want to make sure that those samples are collected properly. Other types of samples can be duodenal aspirates, especially if we're looking for Giardia parasites, and vomitus, although vomitus is very rare. What we want to do is gather a leak-proof container. It should be clean, but it doesn't have to be sterile. Urinalysis cultures or urine cultures do have to be sterile, but stool cultures do not have to be sterile. It doesn't have to be in a sterile container. We want to collect our sample and put it into the container, and we don't want our container to have any contaminants such as soaps, detergent, urine, or toilet water. Soaps, detergents, and toilet water can actually kill fragile parasites, and actually toilet water can contain its own parasites. Okay, we want to culture the sample as soon as possible while those bacteria are still viable. And then if we cannot transfer the samples onto the media as soon as possible, then instead we want to put that stool sample into a holding media such as glycerol phosphate buffer or Carey-Blair media. We want to be mindful of refrigeration as this can harm some pathogens, and often several samples are collected for studies over several days. For example, if we're doing an ova and parasite study, there might be 3 samples collected over 3 days. So let's take a look at containers that are not acceptable for stool culture. It's probably hard to believe, but yes, I have seen stool cultures collected in these types of containers, and these are not acceptable for stool samples. So let's take a look. Test tubes. Good luck trying to get a stool sample in a test tube or a red top tube, but I've seen it happen. It's not the best collection tube for a stool sample. Windex bottles, I have no idea what people are thinking. Ziploc baggies. Oh, and let's get the big bags. Cardboard box, you can see where that would be a problem. Kind of, your stool sample would kind of seep through that. And then finally, this is my personal favorite. Around the holidays, it's fun to get stool samples in a cookie tin. Warm wishes. Now, gram stains on stool samples are usually not performed. They're not helpful because there are many organisms and morphologies that can be seen in a stool sample for gram stain. However, if we're looking for white cells, then a gram stain might be helpful. If we see polymorphonuclear white cells, there may be a bacterial present with our patient. Our patient may have some sort of invasive organism causing them gastroenteritis. If you see mononuclear cells, you might be thinking of parasitic disease. Gram stains may also be useful if we're looking for an overgrowth of staph or yeast. So on the gram stain, if you see a lot of staph or a lot of yeast due to the overuse of antibiotics, this can cause tummy troubles as well. And also gram stains might be useful if we're looking for a specific morphology. For example, Campylobacter has a gram-negative seagull morphology that it's easy to see on gram stain. Bacillus species, on the other hand, you would see large gram-positive rods with spores, and Clostridium perfringens, you would see boxcar-shaped gram-positive rods. Okay, so again, the gram stain is usually not helpful on a stool sample because you're normally going to see a variety of different organism types. However, it is helpful if you're looking for white cells, staph or yeast, or an overgrowth of staph or yeast, or if you're looking for a specific morphology of organism. There are so many organisms and parasites and viruses that cause gastroenteritis. It's actually virtually impossible to screen for all of the gastrointestinal pathogens, so it's really good to have a thorough patient history. So get that good patient history. We want to know what the patient's age and immune status is. If they're very young, they might be more susceptible to E. coli 0157H7. What's their immune status? They might be more susceptible to Cryptosporidium if they have HIV. Have they traveled recently, and where have they traveled to? Have they traveled someplace where traveler's diarrhea is endemic? Or have they traveled somewhere where they might get cholera? What types of foods have they consumed recently? Have they had leftover fried rice or sushi or maybe even oysters? All of those things are important when getting a good patient history so we can help determine the organism causing our patient's gastroenteritis. So here's some common organisms that cause gastroenteritis and what they are associated with usually. Okay, if you are drinking from a mountain stream and that stream has been contaminated by animal or human feces, you could get Giardia lamblia. It's a parasite. If you've ingested foul dairy products such as milk or ice cream, it's possible you have Salmonella or Listeria. If you've traveled to the east or Gulf Coast, it's possible that you have Vibrio species, especially because there is a very pronounced fishing industry there, and people tend to eat shrimp and shellfish on the east or Gulf Coast more than in the Midwest. If our patient has ingested foul eggs or spoiled potato salad, we might be thinking Staphylococcus aureus or Salmonella. If they've ingested buffet fried rice that's been sitting in your refrigerator for a while, it's possible that you have Staph aureus or Bacillus cereus. If you've ingested undercooked hamburger, that is likely to be associated with E. coli 0157H7. Shellfish that's contaminated is associated with Vibrio or parasites. And then finally, if you're an immunosuppressed patient, it's possible that you have Cryptosporidium or Isospora. Now, if you get sick with a gastrointestinal disease, it usually means you've been exposed to a great deal or a high amount of organism. That's your bug load that you've been exposed to, okay? And most organisms require that you've been exposed to several million to 10 billion bugs, okay? Now, in some cases, organism exposures of only around 10 organisms is enough to make you sick, like Shigella species, for example. It doesn't take very many Shigella species to make a person sick and have bloody diarrhea and just generally feel terrible. So how does the pathogen go and colonize the host? First of all, we're usually exposed to the pathogen by ingesting contaminated food or water. The organism has to be able to survive the pH of the stomach and go through the stomach into the small intestine. And often bacteria or parasites go through the stomach because they are trapped inside a piece of cheese or some sort of dairy product like milk. The organism then colonizes the intestine of the host, and then the organism starts to produce toxins in the intestines. These toxins invade the GI mucosa and prevent the normal absorption and secretion of the gastrointestinal tract. This flux, of course, reveals excessive diarrhea, and that diarrhea can lead to dehydration. Our patients may also have abdominal cramping, and they may have fevers as well, and there could even be signs of blood or mucus in the stool. An enterotoxin is a substance found in the gastrointestinal tract that stimulates hypersecretion of fluid and electrolytes, and this overwhelms the absorption capacity of the large intestine, and then they end up getting diarrhea. The organ that is involved is the small intestine. Now, in this type of gastrointestinal illness, white cells and red cells are absent in the stool, and this is not considered an invasive disease. Some types of organisms associated with enterotoxins would be Staphylococcus aureus, Bacillus cereus, Cryptosporidium, Clostridium botulinum, and Vibrio cholera. Other types of organisms that cause gastrointestinal illness are the invasive pathogens. These are ones that actually invade and damage the mucosa of the large intestine. This results in the loss of white cells and red cells. So you're going to be able to see white cells and red cells in the stool. There may also be protein in the stool as well. Inflammation occurs, and occasionally invasion of the bloodstream starts to occur as well. In the case of the invasive pathogenic diseases, the organ that is involved is the large intestine. Some organisms associated with invasiveness would be Yersinia pestis, Yersinia intercolitica, Campylobacter, Shigella, Salmonella, and then a parasite known as Entamoeba histolytica. So the invasive pathogens are the ones that affect the large bowel, and the toxin-producing pathogens are the ones that affect the small bowel. In a large bowel infection, there are small volumes of diarrhea only, but you can see red cells and white cells in the sample. People that have large bowel infections of invasive pathogens have abdominal pain, fever, mild dehydration because they only have small volumes of diarrhea, and there's usually a slow incubation period of 1 to 3 days. And again, some examples of invasive pathogens would be Shigella and Salmonella. People that have a small bowel infection have large volumes of diarrhea, and usually we do not see white cells or red cells in that sample. They will have mild to no abdominal pain, no fever, severe dehydration because of the volumes of diarrhea they're producing, and there's usually a rapid incubation period for these types of pathogens. Usually the incubation period is about 6 to 12 hours. And the symptoms that our patients have are usually from the production of enterotoxins of the organisms causing the disease. Now, some small bowel pathogens would be Staphylococcus aureus, Bacillus cereus, and others. So here's just kind of an initial list of the organisms that cause large bowel infections and ones that cause small bowel infections. The large bowel infections are those infected with the invasive organisms such as Salmonella, Shigella, Yersinia, Campylobacter, Clostridioides difficile, and E. coli 0157. Other ones that you can add to the list would be things like Entamoeba histolytica, which happens to be a parasite. Organisms that cause small bowel pathogens, small bowel infections would be Staphylococcus aureus, Vibrio cholera, some strains of the enterotoxigenic E. coli, and then also Cryptosporidium and Bacillus cereus. Those are not on the list, but you might want to jot those down. If you have a gastrointestinal illness, what is the expected outcome? Usually it is self-limiting and not treated with antibiotics. So Salmonella, Staphylococcus, Campylobacter, all of those are self-limiting. The only time you might see a patient treated with antibiotics is if our patient has Shigella. Usually our patient will have severe dehydration as a consequence of having a gastrointestinal illness, and they may develop what's known as toxic megacolon. Toxic megacolon is a condition when the colon swells and holes start to develop in the colon, so stool actually spills out of the colon and into the abdominal cavity. Patients may also develop Reiter syndrome, which is a reactive type of arthritis. Other diseases associated with gastrointestinal disease would be hemolytic uremic syndrome, and also most people will experience a malabsorption or absorptive surface of the colon that can be damaged or sloughed away. Like I said, most of the time for the gastrointestinal diseases, they are self-limiting, and so the doctor is most likely to let the disease run its course, and they're only going to give you supportive therapy. Now, times where the patient is treated with antibiotics is when they might be immunosuppressed, or if they have been traveling to a developing country, if the patient is experiencing bloody stools, or if they've had diarrhea for an extended period of time, or if the disease requires intravenous rehydration, or if the patient has a fever that has not resolved. These are certain conditions where the patient would be treated possibly with antibiotics, but certainly with antidiarrheals, drugs, and also fluids. If the patient does require intervention, we do want to rehydrate the patient with IV fluids. The patient may be given antidiarrheal medications, and in rare circumstances, antibiotics might be prescribed, especially in the case of Shigella. There's a number of things that we want to do in order to isolate the potential pathogen. For example, we want to make sure that we set up the proper media required for the suspected pathogen. For example, if we suspect our patient has Campylobacter, then we want to set up Campylobacter CVA or Campylobacter blood agar in order to isolate that pathogen. We also want to determine the optimal atmosphere for the pathogen growth. If you're dealing with the Campylobacter, for example, we want to make sure that it gets into a microaerophilic environment. We always want to make sure that the temperature is correct as well. For example, most human pathogens grow at 35 degrees, which is body temperature, but certain organisms like Campylobacter can grow at 42 degrees. We want to determine if an enrichment broth should be included with the culture. So if we suspect our patient doesn't have that much Salmonella or Shigella in their intestinal tract, we could set up a selenite or gram-negative broth. And what that's going to do is increase the numbers of Salmonella and Shigella for purposes of identification. We also want to check with the physician to see if an ova and parasite study needs to be done on our patient. Okay, and then we also want to determine if any type of serology or immunochemistry tests need to be performed to rule out viral infections, such as rotavirus or norovirus. Hepatitis A is another one. We could do a test known as the ELISA test. So there's a number of different stool culture types the physician can order on our patient that has gastroenteritis. Okay, a routine culture is usually what's ordered, and that includes the identification of Salmonella, Shigella, and Campylobacter. We also tend to isolate all the normal enteric flora as well, but we don't identify that. We only look for Salmonella, Shigella, and Campylobacter, which are the three big stool pathogens. Now, if your doctor wants us to look for E. coli 0157H7 or Yersinia or Vibrio, those are special cultures requiring special media, so the doctor would want to make sure to let us know to look for those pathogens. Okay, special media would be CIN. CIN is for the isolation of Yersinia intracolitica. There's also special media for Vibrio cholera, and then McConkey with sorbitol is our best media to isolate E. coli 0157H7. For a routine culture, we're going to set up a blood and then some sort of mildly selective media such as McConkey or EMB. And then we're going to set up a moderately selective media such as hectoenteric, SS, or XLD. And then we want to set up CVA for Campylobacter. And then we also set up either a gram-negative or selenite broth to kind of supercharge those small numbers of Salmonella or Shigella. So again, I have a list here of the invasive organisms. These are the pathogens that actually destroy the intestinal mucosa. And I always tend to think of Shigella, Salmonella, Yersinia intracolitica, and Campylobacter. So let's start by talking about Shigella. If a person gets a Shigella infection, it's because they have ingested contaminated food or water. This is an invasive pathogen where you're going to see red cells and white cells in the stool samples. Patients may be producing Shiga toxins, but it is not considered a toxic strain. Okay, Shigella is communicable and reportable, so we need to call it to the doctor, the epidemiologist at the hospital, and also the state health department. There are 4 common species of Shigella. Shigella sonii is the most common in the United States, while Shigella dysenteriae is the most virulent and dangerous. Okay, Shigella causes dysentery, and the best strategy for isolation is setting up a blood agar plate, a MAC or an EMB, and an HE or XLD, and then some sort of broth, either a gram-negative broth or a selenite broth. This is one of the few bacterial causes of gastroenteritis which may require treatment with antibiotics like ampicillin or tetracycline. Usually symptoms of Shigella develop about 24 to 48 hours after they've eaten. Okay, this can lead to a very serious disease in babies where the toxins or neurotoxins may prevent the anal opening from closing. Every Shigella can be serotyped, and so every Shigella has a scientific name and then also a serotype name. Okay, so Shigella dysenteriae is also known as Shigella group A, and this is the most serious type of Shigella a person can be infected with. Then we have Shigella flexneri, which is group B Shigella. This is the second most common Shigella isolated in the United States. Then we have Shigella boydii, which is known as group C Shigella. And finally, we have the most common Shigella isolated in the United States, which is Shigella sonii, which is also known as group D Shigella. Now, Shigella dysenteriae, flexneri, and boydii are all identical except for serologically. Okay, Shigella sonii, on the other hand, is unique because it's ODC positive and ONPG positive. If our patient has a Salmonella infection, they most likely got the infection from eating contaminated food or water, and that can be food that is contaminated with human or animal feces. Once they've been exposed to salmonella, it usually takes about 1 to 3 days before they start to develop symptoms. They can actually get salmonella from things like raw meat, poultry, milk, and eggs. Person-to-person transmission is common, and salmonella, like Shigella, is communicable and reportable. This may cause gastroenteritis, or it could lead to what's known as typhoid fever, which is also known as enteric fever. Salmonella can also lead to septicemia, or our patient can actually be treated for salmonella and then they can become a carrier of salmonella without any symptoms. Think of Typhoid Mary. She was a carrier of typhoid fever even though she did not have symptoms herself. The best strategy for isolation is to set up a blood agar plate, then a MAC or an EMB, an HE and an XLD, and a broth. Keep in mind, salmonella has a very complex taxonomy with about 2,400 different serotypes. This is almost always a self-limiting disease, but in rare circumstances it does require treatment with ampicillin or chloramphenicol. Antibiotics may actually prolong the illness. Salmonella can survive phagocytosis and go into the bloodstream, gallbladder, and get stuck in the gallbladder as a glob, and then it's constantly being shed in the stool, and that is when our patient is in what's known as a carrier state. So to set up and identify Salmonella and Shigella cultures, we want to set a blood agar and then both a mild and moderate to highly selective media, and then also an enrichment broth for their isolation. We're going to incubate those plates aerobically for 24 hours at 35 degrees Celsius. Then we can subculture our enrichment broths within 24 hours, and we're going to subculture those to the same mild and highly selective medias originally set. Then we can biochemically or metabolically make a presumptive identification. Then we have to serotype it in order to give that information to the state health department. We can set up susceptibility tests as needed. Again, typically Shigella needs susceptibility testing, but Salmonella typically does not. Let's go ahead and take a look at what Salmonella and Shigella will look like on your culture plates. So Salmonella is lactose and sucrose negative. So your MAC organism is going to be colorless. Your EMB organism is going to be colorless. Salmonella on HE is going to be clear green with black centers because it is an H2S producer. And on XLD, Salmonella is going to be clear red with black centers. Now Shigella, on the other hand, it is lactose and sucrose negative as well, but it does not produce H2S. So on MAC and EMB, Shigella is going to be colorless. On HE, Shigella is going to be clear green, and on XLD, our organisms are going to be clear red. Now keep in mind, Shigella sonii is what's known as a slow lactose fermenter. You probably remember that it's OMPG positive. Because Shigella sonii is a slow lactose fermenter, beyond 24 hours, you may see some slight yellow on XLD or salmon on HE because it is fermenting lactose. Okay, anytime we're looking for Salmonella or Shigella, we look at our plates and we always look for lactose sucrose negative colonies. If they're lactose sucrose negative and H2S positive, it could be a Salmonella. If you have lactose sucrose negative colonies elsewhere that are H2S negative, they could be Shigella. So if you take a look at this plate here, you can start to determine which organism is a Salmonella. Okay, so this is a mixed flora sample placed on a hectoenteric agar. Okay, the colonies that are yellow or kind of salmon in color, kind of orangey salmon in color, those are ones that produce lactose and sucrose fermentation. So we would ignore those colonies because that's probably routine normal flora. Now, the Salmonella on this plate is the one that is clear green with a black center. Those are the ones that I would be highly suspicious are Salmonella. So those are the ones that I'm going to set up my metabolic tests on. Here we have an example of a McConkey agar and an example of XLD. You can probably start to figure out which are the Shigella species based on what they look like on those plates. So if you look at the plate on the left, this is a McConkey plate, and you can see there's pink colonies and then colorless colonies. The pink colonies are probably normal flora, like maybe an E. coli or a Citrobacter, and you can see that the colorless colonies are likely to be your Shigella because they are lactose sucrose non-fermenters. If you take a look at the XLD plate on the right, you can see yellow colonies. Well, our yellow colonies are lactose and sucrose fermenters, so that is not Shigella, but you can also see the clear red colonies. Those clear red colonies are the ones that are likely to be Shigella because they are lactose sucrose negative. Setting up a TSI is helpful whenever we are investigating possible Salmonella or Shigella identification. Okay, all of our Salmonellas and Shigellas are going to be alkaline over acid, so they don't ferment lactose or sucrose, so the slant of the TSI is going to stay red or be alkaline. Because they're an enteric, they do ferment glucose, so the butt of the slant is always going to be yellow. Okay, so for Shigella, your slant will be alkaline over acid, and for Salmonella, your slant would be alkaline over acid plus H2S. These are the types of colonies that we're looking for whenever we're dealing with Salmonella or Shigella. Now, there's a few steps we have to go through in order to actually confirm Salmonella or Shigella identity. For example, we need to look at the macroscopic appearance of these colonies and make sure that they fit the colony morphology that they should. So Salmonella and Shigella on blood agar are flat, gray, kind of glidy, and then their carbohydrate fermentation, they're going to be lactose sucrose negative, and Salmonella is H2S positive. Once we've identified our organism macroscopically, or once we have an idea that it's suspicious for Salmonella or Shigella, we can set up our biochemical tests or our metabolic tests. Okay, we can set up a conventional ID including tube schemes, API, Vitek, microscan. Those are all great ways of metabolically testing for Salmonella or Shigella. Finally, the last thing that we have to do to confirm the isolation of Salmonella and Shigella is to serotype the isolate. Okay, we are looking for the serotype of the flagellar and somatic antigens. Now, Campylobacter is the third organism included that we look for in a routine stool culture. If our person has Campylobacter, many of the times they don't even seek physician intervention. Some of their symptoms usually appear 2 to 5 days after eating meat or poultry or milk that has been contaminated with animal feces. Colonies suspicious of being Campylobacter have to be gram stained, and what you're looking for is that gull wing or seagull wing morphology. You may use a prolonged counter stain with safranin in order to get these organisms to really pop microscopically. Okay, they're not fermentative or oxidative. Campylobacter occurs as a gastrointestinal pathogen 7 times more frequently than does Shigella or Salmonella, so it is common. Campylobacter is a frequent colonizer of domestic animals such as house pets, chickens, turkeys, and in contaminated water. These are all ways a person can get exposed to Campylobacter. Some symptoms of Campylobacter would be cramping abdominal pain and bloody diarrhea, chills, and fever. There are many species of Campylobacter, including Campylobacter jejuni, Campylobacter fetus intestinalis, Campylobacter fetus jejuni, Campylobacter coli, which of course means of the colon, and Laurie. Those are our different species of Campylobacter, but by far the most commonly isolated species is Campylobacter jejuni. That's the one that causes gastroenteritis. Okay, Campylobacter is a microaerophilic organism, so it does prefer 85% nitrogen, about 5% oxygen, and 3 to 10% CO2. The optimal growth temperature for Campylobacter is 42 degrees Celsius, and it does require 48 hours of incubation because it is a slow grower. Now, we want to use selective media for the isolation of Campylobacter, so we could use CVA media, or we could use what's known as Campy blood agar. Both of these medias contain vancomycin and other inhibiting substances, so most gram-positive and other gram-negative organisms are inhibited. Anytime you get growth on your CVA or your Campy blood agar plate, you do want to gram stain it and look for that gull wing morphology. Occasionally, other things can grow on that selective media, so you want to be cautious and do a gram stain. Okay, you can treat Campylobacter with erythromycin, but usually this type of disease is self-limiting and you'll get better within a few days. Okay, so you might be asking yourself, well, how do I get better from a gastrointestinal illness? Firstly, you want to drink plenty of fluids and get plenty of rest. You want to stick with bland foods and wash your hands frequently. And then make sure to rehydrate with oral fluids such as Pedialyte. In certain circumstances, you may be given antibiotics, and that's true in the cases of Shigella and Listeria. You want to avoid high-risk foods such as raw fish, shellfish, eggs, sprouts, juices and ciders, deli meats, milk, soft cheeses, and even hot dogs. If you have an organism growing on your CVA plate, the first thing you want to do is gram stain it. If you see a gram-negative rod with a gull wing morphology on selective media, then the next thing you want to do is rule out Campylobacter by doing a catalase and oxidase. Campylobacter jejuni is catalase positive and oxidase positive, and it's also hippurate hydrolysis positive. If we set up a couple discs on our plate, you will see that Campylobacter jejuni is resistant to cephalothin, sensitive to nalidixic acid, and finally, to confirm it, we would serotype. So we always want to serotype any of these pathogens like Salmonella, Shigella, and Campylobacter. So that's it. That's Unit 17, where we start to discuss gastrointestinal tract infections and stool cultures primarily related to Salmonella, Shigella, and Campylobacter. Those are all part of a routine stool culture. Have a good day. Bye.